Namaskaram. Today we are going to see children Hershey's and Chase Experiment. This is one of the unit of molecular basis of inheritance. This is another important topic from NEET Aspect. If you are new to my channel, please go through my entire lecture, and if at all you like the way I explain, please do like and share it amongst your friends and do subscribe the channel. So without wasting any time, let's start with the experiment. Before going on to Hershey's and Chase experiment, you should have a rough idea about what this experiment is about. This experiment is dealing with to find out what is the genetic material. Okay, this experiment was carried out by Hershey and Chase. These are two different scientists. Hershey was a male person and Chase was a female. Okay, so Hershey and Chase together they worked on this experiment in order to find out what is the genetic material. Now this experiment was carried out about 150 years ago approximately in 1952. Okay. So in that knew what was the genetic material in a cell. Right? What is a genetic material? A material which gets carried over or passed on from one generation to another. So before this we have seen two experiments, that is Griffith's experiment and Avery, Macleod and McCarty's experiment. So based on these two experiments, Hershey and Chase performed a wonderful experiment to find out what is the genetic material. It was actually just a confirmation to Griffith's experiment. Before that, I'll tell you that in what Griffith did. What Griffith did was, Griffith used mice and he used the bacteria Streptococcus pneumoniae. Right? So these two organisms were used by Griffith in order to find out that what is the material which goes from the bacteria to the mice and the mice gets killed. So somewhat similar to this, these two people did the experiment. What they took for the experiment was E. coli. This is again a bacteria, Escherichia coli. This bacteria resides in the colon of our intestine. So it is called Escherichia coli. And they used a virus called as Bacteriophage. Understand children, Bacteriophage is not a bacteria, it is a virus. Often children get confused that Bacteriophage is a bacteria. No, it's a virus. But virus, such virus that eats bacteria. Phage meaning what? Phage refers to eat. It is called bacteriophage. So Hershey and Chase, what they did, they used a virus, okay, for infecting E. coli. Right? Like how Griffith used Streptococcus pneumoniae to infect the mice. In a similar manner, they used this virus to infect bacteria, E. coli. And then they found out that what was the material which went from the virus to the bacteria, whether it was DNA or it was protein. Understood? So it's running parallel to Griffith's experiment. We will have to know certain basics about the pattern of infection of this virus to the bacteria, right? How does it infect? Right. Now, there are different shapes of viruses. Okay. Generally, in this case, the bacteriophage is T2 phage, okay, even T2. 2 is an even number. So there are n number of viruses. You have only two genetic material, DNA or RNA. Then in RNA you have single stranded RNA. In DNA you have single stranded DNA and double stranded DNA also. Right? So accordingly these viruses are classified as DNA viruses, RNA viruses. Then in DNA also they are classified as even phages and odd phages. So this is an even bacteriophage. This is a virus or a bacteriophage which eats bacteria and it is designated as T2. Okay. Now let us see how does this virus look like and how does it infect the bacteria. See, this is the head of the virus. This is its middle piece, or you can say the body, actual body of the virus. This is the neck of the virus. And then you have the base portion. That is how it looks like. Okay. Now we are dealing with a bacteriophage which is having genetic material as DNA. This is double-stranded linear DNA acting as the genetic material of this bacteriophage. This is the head of bacteriophage. Okay, like how we have different parts of the body, similarly the virus is having head. This head is often referred to as protein coat. Coat meaning what? Coat meaning protection, a covering. The genetic material of the virus is protected by a protein coat. It's also known as capsid. Now understand, here I am saying protein coat because now we know that the covering of the virus is made up of protein. But actually in that time, 150 years back, we did not know that the head of the virus is made up of protein, right? In this experiment, we are going to see that. Now this is the middle piece, the middle portion. It's called as middle piece. And these are the tail fibers. Meaning with the help of this tail fibers, this virus is going to sit on the bacterial surface and it is going to infect the bacteria. Now simultaneously we'll see how is the E. coli cell. Say it is cocobacillary. It's slightly coccus and it is slightly rod. These lines which I am drawing is the pili, or they are called as sex pili. Okay, with the help of this sex pili, there is exchange of genetic material from one bacterial cell to the other. Okay. And the bacterial cell has got a long filament-like structure called as flagella. Now E. coli is peritrichous. Peritrichous meaning it has got the flagella throughout the periphery. Flagellum is the locomotory part of the bacteria. With the help of flagella, the bacteria can move. Understand, all bacteria do not have flagella, but E. coli does have. Okay. Now in the center, I am going to draw the genetic material of E. coli, which is again a double-stranded, but in this case it is double-stranded circular DNA. Now this virus or this bacteriophage is going to infect the bacteria. Process of infection. Say this is the bacteria. This is the bacterial surface. Okay. Now the first step of infection is called as, okay, meaning now the virus is going to infect the E. coli cell. I am drawing the genetic material with different colors so that there is no confusion. The second step of it is adsorption. Adsorption. Now in chemistry, oftenly we know these terms: absorption, adsorption. Adsorption meaning what? Two unsimilar or two dissimilar surfaces are coming in contact with each other. So here the tail fibers of the virus and the cell wall of the bacteria are going to touch each other. Okay, so it is called adsorption. The diagram will look somewhat like this. Now actually the tail fibers are touching the surface of the cell wall, right? So this is adsorption. In the third phase, this is adsorption. Third step is known as penetration. Now what is getting penetrated into the E. coli cell after infection? It is the genetic material. Now we are doing this experiment, we in the sense Hershey and Chase did this experiment to find out which is that genetic material, whether it is DNA going into the bacterial cell or it is protein going into the bacterial cell. We do not know that whether it is the protein of the virus entering into the cell or the DNA of the virus entering into the cell. That is what we are going to find out. But just now for time being, we will say that the genetic material is penetrating from the virus into the cell, right? So this is the third step, wherein I will show the penetration something is going into the cell, right? And of course you have to draw the genetic material of E. coli also. So understand, this is the genetic material of the bacterial cell and this is some material, genetic material of the virus penetrating into the cell. After penetration, what happens is the game starts. Meaning what? The genetic material of the virus is so intelligent that it will capture over, it will take over the genetic material of the bacterial cell. It will order the E. coli cell that now you synthesize my parts, right? Because we know that the virus needs to multiply. But virus is what? It is not cellular. It's on the borderline of living and non-living. Meaning when the virus is in the air, it is non-living. But when it enters into particular host, it becomes living. Now why does it become living? Because it takes all the material of the host bacterium cell and it synthesizes its own parts. Own parts in the sense its head, its middle piece, its tail fibers, everything is synthesized by using the host machinery, right? So this order is given by the viral genetic material to the bacterial genetic material, right? So after penetration, there is synthesis of viral parts, and after that there is multiplication, because once the viral part starts getting synthesized, the virus will grow, it will multiply, right? And finally there will be bursting of the bacterial cell and release of viruses. Here, what will happen is, now the genetic material has entered into the cell. This genetic material will command the bacterial genetic material to make its own part. Here you will find synthesis of head somewhere, synthesis of middle piece somewhere, synthesis of tail fibers, right? All these are synthesized individually and separately. Why do they start synthesis? Because the viral genetic material has now ordered the bacterial genetic material that now you make my parts, you don't do your job. Understood? It's like we know that in a cell DNA takes over, right? In every cell DNA is the master commanding molecule. It will tell the cell that what to synthesize, what not to synthesize, when to synthesize, when not to synthesize, when the cell should divide, right? So this all is governed by the DNA of the cell, whether it is bacterial cell, plant cell or animal cell. But after the entry of this genetic material of virus, this bacterial cell has now no identity. Now it will work according to the command given by the viral genetic material. And so the bacteria stops synthesizing its own material and it starts synthesizing viral parts, right? And then slowly what happens is, this virus starts taking charge over the bacteria and then it organizes its all part. That means it assembles its all parts and it starts multiplying inside the bacterial cell. You can say that now the virus has become viral, right? Because it is multiplying. After multiplication, finally what happens is, the bacterial cell is now so overloaded with the virus that it finally breaks open, right? It bursts and all the viral particles now come out of the bacterial cell. They are ready again for infection to new bacterial cells. Understood? So the viruses which are released from the host bacterium, now each virus is capable of infecting again a new bacteria. This is how the multiplication goes on, right? So this is a general method how the virus multiplies within the bacterial cell. Now this method is called as transduction. Transduction meaning what? Infection or transformation, you can say that passing over the genetic material from one cell to the other cell via the medium of viruses. Okay, this is known as transduction. In order to understand Hershey and Chase experiment, you must know that how does the virus attack the bacteria, how does it penetrates its genetic material, how it orders the bacterium cell to make its viral parts, and finally how the viruses are released from the bacterial cell. This process, if you understand, then Hershey and Chase experiment becomes easy to. Now Hershey and Chase, they used two radioisotopes. Okay, they took the help of radioactivity to detect what? To detect that what goes into the bacterial cell. whether it is protein or whether it is DNA, right? We have to find out that what is going from virus to bacteria, whether it is DNA or protein. Now we know that bacteria is too small. You cannot see with the naked eye. Virus is even smaller than the bacterial cell. So it's not possible to detect what is going or what inside the cell. So in order to make it visible, they took help of the radioactive isotopes. Right? We know in chemistry what is radioactive isotope. You can say a heavy form of an atom of a particular element. Now we know in proteins we have sulfur, right? What are proteins made up of? They are made up of amino acids, and amino acids have got carbon, hydrogen, oxygen, nitrogen. Apart from that, they do also have sulfur, right? But sulfur is absent in what? Sulfur is absent in DNA, right? So for proteins they used sulfur, right? And for DNA they used phosphorus, because we know that phosphorus is present in phosphates of DNA. Deoxyribonucleic acids, they are acids, right? And acid part comes from the phosphate of DNA. So they took phosphorus for labeling DNA. And we know that phosphorus is absent in proteins. Now, what is the radioactive element? Sulfur. Now, sulfur is S35 and phosphorus is P32. Right? There shouldn't be any jumbling between what is the atomic mass of sulfur and phosphorus taken. Very simple way to memorize is what? How do we learn P, Q, R, S? P comes first, S comes later, right? This is how I have memorized. Phosphorus 32, sulfur 35. So 32 is smaller than 35, P comes before sulfur. So they used these two isotopes, that is S35 and P32, to label, to radioactively label proteins and DNA. Now what is this term I am using? Radioactive labeling. Labeling meaning what? Labeling meaning designation, right? You label your notebook with your name, with your standard, right? So that your book doesn't get lost. In the same way, radioactive labeling meaning we are incorporating these radioactive isotopes in proteins and DNA, so that we can see that which kind of element or which kind of isotope is entering into the bacterial cell, right? Whether it is S35 entering into the bacterial cell or whether it is P32 entering into the bacterial cell after the infection of that virus. Okay, so what we have to do, whom do we have to radio label? We'll have to radio label the viruses, or you can call them as bacteriophage. I'm writing short form bphage. Okay, because if we label this bacteriophage with S35 and P32, right, we will able to detect that which part is entering into the bacterium. Okay, whether it is S35 entering into the bacterium or whether it is P32 entering into the bacterium. Understood? So, if S35 is entering into the bacterium, the answer is clear cut that S is present where? It is present only in proteins. It is not present in DNA. That means proteins are entering into the cell, and so protein is the genetic material. But if P32 is entering into the cell from the virus, then it is obvious that DNA is the genetic material which is entering into the bacterial cell. Understood? So this was a very simple clear-cut logic which Hershey and Chase used. What they did was they radioactively labeled the viruses, and then after radioactively labeling the viruses, then they did the infection on the bacterial cell. Okay, so we are going to see the entire process. step by step. Not by just using the heavy isotope, radioactive isotope, will it become clear that which is the genetic material. After radioactively labeling the virus with that radioactive label, then we will allow that virus to infect into the bacterium, and then we are going to expose mixture to X-ray diffraction. Okay, meaning suppose if I am having a solution, right, some X solution like this, and in this solution I am having this radioactive phosphorus. Okay, and in other tube, say I have a mixture, and in this I am having S35, right? Now what I'll do is I will expose this both tubes to X-ray diffraction. Okay, I will allow the X-rays to pass through this tube containing this mixture which is having radioactive element in it. After that, after exposure to X-rays, what will happen is there will be some rays coming out of the tube. When you are making incident some rays through a solution, then the rays which are emitting out will be collected, right? So these rays coming out from the tube, they are allowed to pass through a detector. Okay, so you have some radioactive detector. What will it do? It will detect the rays coming from this tube, right? Then these rays will be analyzed by the detector. It's just like how you have a electrocardiogram, ECG. So that will tell you the impulses, PQRST, the wave of heart, how it is pumping, right? In the same manner, these rays which come out after penetrating through the tube, these rays are collected, they are detected, they are analyzed, and then they are represented in the form of radiogram. Okay, this is called as radiogram, or it's called as autoradiogram, right? And this process is known as autoradiography. Okay, why graphy? Graphy meaning it is represented in graphical form. The result is detected in graphical form, so graphy. Autoradio because you are using the detector called as autoradiographer, okay, or autoradiogram you can say. So this is how they used this radioactivity. Now what happens is when you pass these X-rays through the solution containing radioactive isotopes, then there will be fluorescence. Okay, there will be fluorescence. The term used is inflorescence. Inflorescence meaning what? They will shine, they will bright, they will illuminate. Okay, the radioactive isotopes will show fluorescence. It's just the fluorescent colors, how they look. Fluorescent green, fluorescent yellow, they are very bright. So you can easily detect the fluorescent colors from the other normal colors, warm colors. In the same way, after passing the X-rays through this radioactive solution, there will be inflorescence. This inflorescence will be detected by the radioautography, right, or autoradiography you can say, by the radiogram, and it will be resulted in the form of graph. This is how you can detect that what kind of radioactive isotope is present, in what concentration it is present in that solution. Okay, this basic principle of autoradiography you have to understand in order to actually see the experiment. Okay, we have seen the basics, that how the virus infects the bacteria, first thing. Second thing, that how by using radioactivity we can detect the inflorescence. Okay, so these are two important aspects before moving on to actual experiment. Say I am having this container, right, which is containing a medium. Okay, now in biology what do we mean by medium? It's not the medium in physics, okay, that air medium, water medium. The medium in biology refers to the food material, okay, the food which we are providing for the virus to grow, or the food that we are using for the bacteria to grow. Now we know that viruses are acellular. They cannot live outside. They need a host cell, right? So we will require E. coli cells in order to grow viruses, isn't it? Because viruses cannot multiply outside the cell. Just now we saw that how that bacteriophage infects the bacterium, it multiplies within the host cell. So if we have to observe viruses, if we have to grow and study viruses, we will require some cells, okay, living cells for infection. So here we are using E. coli. So suppose this is my E. coli cell, right? And I am infecting this E. coli cell with the virus, right? And say this is the genetic material inside the virus. Now, what we are going to do is we are going to allow this virus to infect the bacterium inside the medium containing what? Containing, say, sulfate. And sulfate which is having sulfur with S35 isotope and not the normal isotope. We are using heavy isotope, right? Now what will happen is you will allow this virus to grow in this medium for several generations. Okay. For several generations, we will keep that virus infecting the bacterial cells, okay, in a medium containing S35. So eventually what will happen is this sulfur, which is heavy isotope, this sulfur will now enter into what? This sulfur will now enter into the protein of virus. Okay, because we know that viruses are also made up of what? They are also made up of DNA and proteins. You are getting my point, right? So viruses are also made up of DNA and proteins. So we are growing those viruses in a medium containing sulfur which is of S35 for several generations. Why several generations? Because in one generation the sulfur is not going to go into that virus protein, right? It will take time, right, to acclimatize. So for several generations you will allow that virus to infect the E. coli in a medium containing heavy sulfur, right? This is one thing. Now after few generations, if you remove this virus and if you check, okay, if you check for the protein content, you will find that the proteins of the virus have got S35 in them. Virus now have got S35 instead of the normal sulfur. This you can check by the protein. Now we know that the protein gets incorporated into the protein coat, capsid. Okay, this is the head of the virus known as capsid, which is made up of protein. Now this protein we have labeled with what? S35. That means our S35 has gone where? Our S35 has gone into the head of the virus. Understood? This was one experiment which they did. In the second experiment what they did, parallel experiment, that again they took a container containing a medium. In this medium they added phosphates, right? Now this phosphates had an isotope P32 instead of the normal phosphorus, right? So they added the radioactive isotope and they carried out the same experiment, that you have the bacterial cell and you have the bacteriophage infecting the virus, and say this is the genetic material of the virus, right? So for several generations they allowed this virus to infect the bacteria in a medium containing radioactive phosphorus. Again they followed the same procedure. After few generations they again isolated this virus and they detected for radioactivity, right? So the virus had radioactively labeled DNA. What? In the DNA of the virus they found phosphorus which was P32, and here they found a protein which was S35, right? Because we know that sulfur is present in protein and not in DNA. So this S35 will go into the protein coat and not the DNA of the virus. We also know that phosphorus is present in DNA. It is absent in proteins. So this phosphorus will go into the DNA of the virus and not the protein, right? So now what is the case is that we have two things. One is what? Radioactively labeled protein. We have now two types of viruses. One virus which is having radioactively labeled protein and another virus which is having radioactively labeled DNA. Right? So once this is done, now the experiment begins. What we are going to infect this radioactively labeled protein and DNA to the bacterial cell. Right? So let us see how it goes. So deliberately what I will do is, for radioactively labeled portion, I will take red color so that you can understand that this is the radioactively labeled part. I will have two types of viruses. In first virus, I will have the outer part which is radioactively labeled. Now red color I am designating because it is radioactively labeled, right? So here I am having radioactively labeled protein which is having sulfur, right? S35. And here I am having another bacteriophage, right? Another virus which is having radioactively labeled DNA. These two viruses were allowed to infect the bacteria. So here this is P32 in the DNA and this is S35 in the protein coat of the virus. Now these viruses were allowed to infect. After infection, what was done was, first step was, of course, step 1 was what? Step 1 was infection. We have already seen that how the virus infects the bacterial cell, right? Now second step was what? After infection, allowing some time for the virus to infect the bacteria. After some time, then these tubes were allowed to centrifuge. Say I am having a mixture over here which is containing what? Which is containing the bacteria also and which is also containing the virus, right? So I am having two types of test tubes, you can say. In one test tube, what is there? In one test tube, I am having radioactively labeled protein coat, and in other test tube, I am having what? In other test tube, I am having a virus which is having a DNA which is radioactively labeled. So in one tube, I am having virus, protein coat is labeled with radioactive label, and here I am having a virus whose DNA is radioactively labeled, right? Now both of these tubes I am going to allow to blend. Okay, blend meaning what? How you blend in a mixer grinder. So in the same way we are allowing these two test tubes to undergo blending. Blending meaning what? You rigorously shake the test tube. Rigorously meaning with a little force. So when you shake these tubes with force, what will happen is the part which is not going to go into the cell will get separated. Okay. Infection we have already done. The bacteriophage has infected the bacteria. The genetic material has already entered into the cell. Right. Now the part which is not entered into the cell, we need to remove it. Right. So that part is removed. Okay. And then the third step is centrifugation. After shaking, you allow it to centrifuge.
Now what is centrifugation? Centrifugation simply means rotating the tubes, okay, in a instrument called as centrifuge
at one particular speed, that is called as rotations per minute. See, there is an instrument called as centrifuge wherein you can insert the tubes. Okay, these tubes you can place in that instrument, you can close the lid and you allow those tubes to rotate at a very high speed. okay, at a particular speed, that is called rotations per minute. This is called as centrifugation because it is working on the principle of centrifugal force. We know in physics there are two forces, centripetal force and centrifugal force. Centripetal force is towards center of the circle and centrifugal force is away from the center of the circle. So when you place these tubes in that centrifuge, the tubes will rotate at high speed. Why are we rotating? So that the material which is not entering into the cell, bacterial cell, is removed out. Okay, like it's suppose this is the virus, right? And say this is the bacterium. And now say the genetic material has already entered into the cell. So we don't want the rest of the thing. Now what is remaining is just this part. Right? So this is the head, this is the middle piece, these are the tail fibers. The genetic material has already entered into the cell. What we are concerned with is only this part because we want to see that what is entering into the bacterial cell and this we are going to detect by radioactivity. So we don't need this part. So in order to remove this part, the tubes are placed in centrifuge and they are rotated at high speed. Okay. After centrifugation, what you get is you get two things. One is precipitate and the other is supernatant. Suppose I am having a test tube, okay, and in a test tube I am having some muddy water, right? And if I allow that muddy water to centrifuge at high speed and then allow it to settle, what will happen? You know that the mud will get accumulated at the bottom. And what will remain up is the liquid which you call it as supernatant. And this is called as precipitate. So in this experiment also, after carrying out centrifugation, you will get two parts, a liquid which floats above, that is called supernatant, and the solid material which gets settled down is called as the precipitate. Right? Okay, just now we saw. So in the test tube, they had this virus whose head was radioactive, right? And E. coli was lying inside the cell which had the genetic material, its own genetic material. Now in this case, we must understand that this is S35 which is infecting the bacterial cell. After allowing centrifugation, what you will get to see is, right, the head part will go away because we are shaking the tubes at high speed, right? And in the E. coli cell, what you will have is its own DNA, right? Which I have shown with blue color. So this is bacterial DNA plus this DNA which has entered from the virus, okay, into the bacterial cell, right? This will be the condition that this viral DNA has entered into the bacterial cell which I have shown over here, and this part which is of no use now will remain outside, okay, after centrifugation. So I will show it in the form of test tube. Get a precipitate over here, right? So this is your PPT, precipitate, and you have this viral head lying above in the super. This I will say as supernatant part, and this is the precipitate. Now after getting the supernatant and precipitate, we are going to allow this to pass through X-ray, right? And we are going to see the radioactivity, that where does the radioactivity lie, whether it lies in the precipitate or whether it lies in the supernatant, right? So in the precipitate what we are having is E. coli, bacterial cell, right? Now here what happens is the fluorescence is not seen in the precipitate. We are expected that the fluorescence should be in the E. coli cell, in the precipitate, right? What material is going from the virus into the E. coli cell? So we are following radioactivity. But in the first case, what was seen was the radioactivity was shown over here. This part was showing inflorescence, meaning it was shining. after passing through X-ray detector. Okay. But this part showed no inflorescence. This means what? It means that the inflorescence part, that means S sulfur, is remaining outside only. It has not entered into the bacterial cell. So this is a clear-cut understanding that proteins are not going into the cell. Okay, proteins are not entering into the E. coli cell. Understood? Because we have used what first? We have used radioactive S35, and this S35 is in the head portion of the virus. Okay, after infection and after allowing to go under autoradiography, the fluorescence or the inflorescence was seen in the head part, in the supernatant part. It was not seen in the precipitate part where E. coli cells were there. That means the head part or the inflorescence part, or you can say that S sulfur has not entered into the E. coli cell. Now S35 has not entered into the cell. It means what? Proteins have not entered into the E. coli cell. So the answer is straightforward that DNA is the genetic material which is entering into the cell. This was first very clear, but yet they did second experiment wherein the condition was reversed. What? Now they use radioactive phosphorus instead of sulfur. So again, I am having a tube, a beaker containing a medium wherein E. coli cell is there, right? E. coli is having its own DNA, right? And now here they are infecting this E. coli cell with bacteriophage, which is having radioactively labeled DNA, that is P32, right? The same procedure was followed. This is E. coli cell, right? Now in the test tube, you will have two things. One is precipitate, which has settled down, and the supernatant will have the head of the bacteriophage, right? Now here, when you pass this test tube to X-ray, what you get to see is, after blending, what will happen is, after blending, this bacteriophage head will remain in the supernatant. Okay, so this is your supernatant, and what will remain in the test tube. See, this is your PPT, and this PPT is showing inflorescence. This is your PPT, and this PPT is showing inflorescence, right? Meaning it is shining. Okay, when you allow this to pass through X-ray, then this portion is shining. Okay, it is showing inflorescence, and the supernatant part is not showing any inflorescence. Here there is no inflorescence. So this tells you what? That the DNA which was used, okay, this DNA was labeled with P32, right? Now this DNA has penetrated from the penetrated into the bacterial cell. Okay, here it has entered into the bacterial cell, and so the fluorescence was seen in the precipitate and not in the supernatant. This was a very simple experiment done by Hershey and Chase, wherein they used two radioactive isotopes. One was S35. Okay, S35 got incorporated into the head, which did not show inflorescence, and P32, that is DNA, it got incorporated into the E. coli cell in the precipitate, so it showed inflorescence. So this is a clear-cut understanding that DNA goes from the bacteriophage into the E. coli cell. So DNA is the genetic material wherein they use P32 and S35, right? So this was an unequivocal proof that DNA is the genetic material which goes from the bacteriophage to the E. coli cell. Just like in Griffith's experiment, we saw that DNA goes from the Streptococcus pneumoniae into the mice. That is why the mice suffered from pneumonia and it died. So it's a parallel experiment running to the Griffith's experiment. With this background of Hershey and Chase experiment, we will solve some MCQs related to it. Okay. First MCQ is statement 1 and statement 2 are given. You have to find out the four options. Statement 1 and statement 2 both are correct. Statement 1 is correct, statement 2 is incorrect. The third case, statement 1 is wrong, statement 2 is correct, and the last thing, both the statements are wrong, right? So first question is, there are two statements. Unequivocal proof that DNA is the genetic material which came from the experiment of Hershey's and Chase. Second statement, both of them worked in order to find out whether it was DNA or proteins from the viruses that entered into the bacteria. Both of the statements are true, okay? Because this was an unequivocal proof that DNA is the genetic material, and this was proved by whom? Hershey and Chase. It was a confirmation to Griffith's experiment and Avery, Macleod and McCarty's experiment. So statement 1 is correct. Statements 2, they worked on what? Whether it was DNA or proteins from the viruses that entered into the bacteria. So we just now saw the experiment that they were working to find out what? Whether it is DNA or proteins which is entering into the bacterial cell. So this statement is also correct. So accordingly, other four options will be given. First option would be what? Statement 1 and statement 2 both are correct. Second option, statement 1 is correct, statement 2 is incorrect. Third option will be statement 1 is incorrect, statement 2 is correct. And last option will be statement 1 and statement 2 both are incorrect. They are asking in this question, number of histone molecules in each nucleosome code is. Options are 14, 12, 10 and 8. Now we know that histone molecules are an octamer, okay? They are basically an octamer of four types of histone proteins, that is H2A, H2B, H3 and H4. These are four histone types of proteins, and each type we are having as 2 molecules. So that makes up 2 plus 2 plus 2 plus 2, that is 8, right? So answer is straightforward that it is 8. Here they are mentioning it is core, right? So we know that how is the nucleosome? See, you have this octamer 1, 2, 3, 4, 5, 6, 7, 8, and you have the DNA wounding by 1 and 3/4 turn, 1 and 3/4 turn around this core molecule of histone. And then of course you have linker DNA, and then you have another nucleosome like this, okay? So this is your core and this is your linker DNA. Now linker DNA has got H1 molecule, okay? So H1 histone is present outside the nucleosome core. In the core what you are having is just H2A, H2B, H3 and H4, and you are having 2 molecules of each. So read the question properly that they are mentioning nucleosome core and not just nucleosome. Then the number would have been 10 because here also you will have linker DNA, here also you will have linker DNA, here also you will have H1 molecule, here also you will have H1 molecule, right? So check the question first and then jump to conclusion. Which are common for both? Now the question is, which are common for both DNA and RNA? So they have given some nucleotide or units: cytosine, thymine, uracil, adenine, guanine, ribose and phosphate. So we have to see that out of these, which are common to both, I mean which are present in DNA as well as in RNA. Let us check out cytosine. Cytosine is present in DNA as well as RNA. Thymine, no. because thymine is present in DNA, it is absent in RNA. Uracil, no, because uracil is present in RNA, it is absent in DNA. Adenine, yes, because adenine is found in DNA as well as RNA. Guanine, it is also found in both. Ribose, no, ribose is a sugar which is found only in RNA and not DNA. And phosphates, yes, because phosphates are present in DNA as well as RNA. So if you calculate 1, 2, 3, 4, your answer is what? Option 1. Right? Next MCQ. Now here the question is, eukaryotic genome differs from prokaryotic genome. Eukaryotic meaning all higher organisms and prokaryotic meaning bacteria. So you can say that the eukaryotic genome differs from the bacterial genome first. In what? DNA is complexed with histones in eukaryotes. Now, is DNA complexed with histones in prokaryotes? Are histones present in prokaryotes? No. Right? Histone proteins are found in eukaryotes. And in prokaryotes, we have similar proteins called as polyamines. Okay? So, polyamines are present in prokaryotes, but histones are absent in prokaryotes. So, this is not true. Second, DNA is circular and double-stranded in prokaryotes. Okay? So DNA is circular and it is double-stranded in prokaryotes. This is true. But what we have to find out is how eukaryotic genome is different from prokaryotic genome. Third answer is repetitive sequences are present in eukaryotes. Yes, this is true. See, because in eukaryotes you have a DNA, right? Suppose this is a long strand of DNA. So you will have portions which are common and which will go on repeating. Say suppose this red color I have shown, then the red color part of the DNA is signifying what? That certain sequences of DNA are repeating at some intervals. Okay, so these repetitive sequences are found in eukaryotic DNA. They are absent in prokaryotic DNA, right? And genes in eukaryotes are organized into, we get to see operons in bacterial cell also as well as eukaryotic cell also. So this cannot be difference. Okay. DNA is circular in double-stranded prokaryotes. DNA in eukaryotes is also double-stranded. So again, this cannot be the difference. We have to find out that how eukaryotic genome is unique from the prokaryotic genome. The question is, phosphodiester bonds are present in ATP, ADP, GDP, none. Okay. ATP stands for what? Adenosine triphosphate, adenosine diphosphate, guanosine diphosphate, and none. Now we know that phosphoester bonds are present in every nucleotide. So what is one nucleotide? We know that you have a sugar like this, you have nitrogen base over here, and you have phosphate attached over here. So the bond between sugar and phosphate is called as phosphoester bond, right? So this is your one nucleotide. Similarly, you will have another nucleotide over here, second nucleotide. In a DNA strand which is double-stranded, we know that you have complementary base pairs on the two strands. Complementary meaning what? Here if I am having adenine, here I will have thymine. Here if I am having guanine, here I will have cytosine. Here if I am having cytosine, I will have guanine. Here if I am having thymine, I will have adenine. Why? Because A always pairs with T and G always pairs with C with double and triple bond respectively. Right? So this is complementary base pairing in the two strands. So this is my one nucleotide and I'll have second nucleotide, and similarly I'll have n number of nucleotides. So the phosphodiester bond is present between two nucleotides. Here I'll have phosphodiester bond between first nucleotide and second nucleotide, between second and third, and so on, right? So phosphodiester bond will require what? It will require two nucleotides at least, right? So there is one phosphodiester bond between two nucleotides. But here, everywhere they have just mentioned one nucleotide, ATP. They have not mentioned that ATP is binding with some other nucleotide. Here also they have shown ADP. ADP is not joining with any of the other nucleotide. GDP also they have shown it alone, right? So your answer will be none. Why? Because there is no other nucleotide present here. Phosphodiester bond is present in ATP, only ATP? No, it is phosphoester bond present in only one nucleotide. Now they have asked that what is the genetic material in bacteria? Options given are linear DNA with histones, double-stranded DNA without histones, linear DNA with histones, and circular DNA with histones. Now, first of all, genetic material in bacteria. Bacteria will not have histones, no histones, right? This you must understand that bacteria will not have histones in them. They have polyamines. So, wherever there is histones, you will cancel that. So, with histones, cancel. With histones, cancel. With histone, cancel. What is remaining is without histones. Right? So circular DNA. Yes, bacterial DNA is circular. We know that. Bacterial DNA is circular without histones. Right? So answer is straight.
Now the question is, a sequence of DNA molecule contains 20,000 base pairs. Now they have mentioned here that it is base pairs. So we know that it is a linear double-stranded DNA. Because if you are having double-stranded DNA only and only, then you will have these base pairs, right? You cannot have base pairs in single-stranded DNA. How can you have? You need someone to pair with, right? So in single-stranded DNA, you will not have base pairs, okay? So base pairs, I can say, will be absent in single-stranded DNA. In double-stranded DNA, I will have base pairs, right? This is pretty clear. Now, how many base pairs are there? 20,000. So, 1, 2, 3, like this, 20,000 base pairs are there. That means 20,000 bases or nucleotides are present in one strand, and obviously 20,000 bases will be present in the other strand. They are asking what is the number of phosphates in the DNA molecule, meaning in this both double-stranded DNA molecule, what is the total number of phosphates? Now we know that in one nucleotide, we are having one phosphate, right? Say this is the sugar, nitrogen base, and here I am having phosphate 1, phosphate 2, phosphate 3, right? So this is, say, suppose my ATP, adenosine triphosphate. This is one nucleotide having 3 phosphates, 1 sugar and 1 nitrogen base, right? But if we are not considering 3 phosphates, say I am just considering 1 phosphate, then in one nucleotide I will at least have 1 phosphate, right? So if I am having 20,000 plus 20,000 is how much? 40,000. So if I am having 40,000 nucleotides, then I have at least 40,000 phosphates in it, right? If I am considering mononucleotides, adenosine monophosphate, not triphosphate or diphosphate. If I am considering monophosphate, I will at least have 1 phosphate. So accordingly, if I am having 40,000 nucleotides, I will have 40,000 phosphates present in it.
Now the question is, in Griffith's experiment, which of the following when injected into mice will result in death of the mice? Okay. First option is R live strain. If you are injecting R type of bacteria of Streptococcus pneumoniae into the bacterial cell, the mice will die or survive? It will survive. Why? Because the rough strain does not have a capsule which is pathogenic, right? Or which is virulent. Heat killed S strain. Now, if you just inject heat killed S strain, still the mice is going to survive. Why? Because the bacteria are dead. In the third case, living R strain, meaning living R bacteria and heat killed S strain. If you inject this both, then the mice will die. Mice will die. Why? Because of transformation. We know the Griffith's experiment. And last option is both 1 and 3. 1 and 3, no, this cannot be the case, right? So our answer is the third option. I hope you have understood the Hershey's and Chase experiment. If at all you have some queries, you can put down in the comment section. Okay. Thank you so much for listening to the talk.